All right. Yeah. I'll just start writing it just in case. Let me know if I'm like out of frame.
Yeah, no, you're good. Your head is in the way.
It is?
Yeah.
Can you see this? Or
Can you reset I? Or do yeah, it's better. Now, do you want the Terminators on?
oh, you're right. You see that? All right, cool. Okay. Anybody have any questions? No? Okay.
Nope.
Cool. So we'll go over lac operon. Basically, the first thing you should know is from the name, we can break down the name to two parts, right? Lac being what it does it's an operon specifically for lactose. That's where the lac comes from. And an operon is basically like biological term for a set of genes that are controlled by or a set of similar genes that are controlled by a similar or by one promoter. So as you guys can see here, there is one promoter. There's one promoter that controls this set of similar genes, and it's pretty useful because if there's a promoter that controls a similar set of genes that all work together for one purpose, then you don't have to get transcription to happen over there and then over here and then over there. You can just have it all occur right here, and that way you can get all three genes expressed for a certain purpose, and you don't have to like scramble to find them all in the DNA. Like they're all together, so it's easier for process to occur and all that stuff. So if you guys have any guesses on what the Z gene codes for, and what the Y gene codes for, and what the A gene codes for, post them in the chat. Let me know what you guys think. But before that, we'll talk about the repressor. Can you see that? Yeah.
I see your color coordinating today. For one.
Is it still in frame?
Yeah, it will be. Your head's in there.
Okay.
It's good now.
Okay, cool.
It looks like TX8.
TXA?
TX8. What is that?
Transcription. Yeah, translation. All right, cool. So when it comes to the repressor, it is not considered a part of this operon. And that makes sense because, like we said in the beginning, an operon is defined as a similar set of genes under one promoter, right? And so this promoter is for these structural or these genes, and the repressor has its own promoter, which is important because this repressor is going to be expressed no matter what. So what we have is that we have this gene called LAC-I gene, which is a regulatory gene. All right, this regulatory gene codes for a repressor, right? And when I say codes for, I mean that it has the genetic material that once it's transcribed and then translated into a protein, it creates this repressor that you see here. And so basically in the scenario, well, actually, let's take a step back. So the lac operon, like I said in the beginning, or like I mentioned in the beginning, has to do with lactose, and it's a bunch of genes that have to deal with the actual breakdown of lactose, right? So if there is no lactose present, then if there's no lactose present, then this operon should be off, right? Because if you have no lactose and the purpose of this operon is to break down lactose and there's no lactose, why would you want it on? And so you want it to be off when there's no lactose. And so that's the case we're looking at right now. And the reason why you want it off is because you don't want to waste energy transcribing these and then translating these if there's no purpose of having it, if there's no lactose to break down. So we're going to have this repressor. This is when there's no lactose present. We're going to have this repressor, and it's going to come to the operator. And it's going to bind to the operator. I drew it kind of like a puzzle piece because when there's nothing, when there's no lactose present, this will be expressed in a way that it's active. Right now it's active. That means that it's active. That means that it can bind to the operator and then stop transcription for this operon. And the way you can kind of think of it is that the the way you can kind of think of it is this RNA polymerase, right? It's going to bind to the promoter and it's going to want to transcribe these genes, right? But as soon as it gets to this promoter right here and starts going downstream to transcribe, it'll reach this obstacle in the road, and it'll realize that maybe I shouldn't transcribe these, and so it'll be like, okay, I'm good, and it'll detach from the promoter and leave, right? So leaving these genes untranscribed. So that's the case when there's no lactose. Yeah. Oh, no lactose. Yeah. So I'll give you guys like a minute to digest that, and put in the chat any questions that you guys might have, and then from there we'll move on to the case where there is lactose. All right, so we are going to move on now to the lactose present condition. Okay, erase. I didn't have to erase that. This paper is really good.
Yeah.
No, it's not color-coded.
There's a blue.
There is?
Where? No. Did you have one?
That's fine. There we go. All right. So now we're going to talk about in the case where there is lactose present. And remember, because this lac operon, the purpose of it is to break down lactose. Then you already know before you even think about what's going on that it's supposed to be on, right? So this will still create the repressor. However, if you didn't notice before, I draw this repressor with a little indentation, and what goes in there is actually a molecule called allolactose. Allolactose is kind of a different form of lactose, but it serves as the thing that binds to the repressor and takes it from its active to its inactive form. So as you can see, the reason why this one's inactive is it's not this nice puzzle piece anymore. It's kind of bent. And so when it tries to go bind to the operator, it won't be able to because it's kind of like this now. So the function of that is now you have this operator, or now you have this operon that will be on because there's no repressor telling the RNA polymerase that when it binds to the promoter, that it can't go and transcribe all these other genes right here. And so now we can talk about what these genes are. LAC-Z, that's galactosidase. Permease. Transincitylase. So as you can all tell from the suffixes, they're all enzymes, right? Because they're all end with the ACE suffix. This one right here is beta galactosidase, right? So basically, this enzyme is going to be responsible for breaking down the lactose that we are introduced into them, or that's introduced from the outside. But before that can happen, we need to bring this lactose molecule that's outside the cell into the cell, right? So that's where permease comes in, and permease helps lactose that's outside the cell come in. And then the last one, which is kind of unknown of what it does, is transincitylase. That's the LAC-A gene. People don't really know what it's for. We know it's important because it's in the operon, and so it has to serve a specific purpose. But most people don't know what it is. Yeah. So yeah, those three working together, they allow for lactose outside the cell to come in, be broken down, and used. And so the reason why that's important is because the reason why that's important is because lactose is a disaccharide, and it's made out of glucose and galactose, right? But out of these two, your body loves using glucose to break down and create energy and all that stuff. So your body wants this glucose that's within lactose. But before it can get glucose, it needs to break this bond right here and get rid of that galactose and use that glucose that it wants. Like that. So that's going to be important when we talk about with the presence of glucose as well. What else can I talk about? That's about it. Yeah. So in summary, when there's lactose present, right, the operon is going to be on because you know that the function of this operon is to break down lactose, right? So you want the operon to be on so that the lactose that is present can be taken into the cell and broken down for that glucose that the body needs as an energy source. And the reason why it's on is because once lactose is brought into the cell, some of it is converted into allolactose, which is the molecule that turns the repressor from its active to its inactive state. And that means that it's not able to bind to the operator anymore. And therefore, the RNA polymerase is able to bind to the promoter, and the RNA polymerase doesn't have anything impeding its path. And so it's able to successfully transcribe LAC-Z, which codes for beta-galactosidase, LAC-Y, which codes for permease, and LAC-A, which codes for transacetylase. And each one serves a purpose. Like I said, beta-galactosidase is to break this bond right here. Permease is the one that will take this molecule from outside the cell into the cell. And then transacetylase, nobody knows. But if you have any theories, put them in the comments. And yeah, that's the summary. And then most importantly, the operon will be on when lactose is present. So yeah, I'll give you a minute to, if you have any questions about this at all, anything at all, or the one before when there was no lactose present, make sure to put it in the comments. Did you have a question, Derek?
You forgot about CAP. Deoxycyclase CAP activation.
I was going to talk about that when we talk about glucose. Or should I talk about it in this kind?
I think you're talking about it now because like on.
You're right. You're right. Okay. So, like Derek pointed out, there's also another thing that we need to talk about, and that's CAP. So, look at the race. So, like Derek pointed out, when we're talking about this process going on, we also have, and in addition to the repressor, we also have a transcription factor that's working as well. So, yeah. So, like Derek pointed out, transcription factor are really not a part of prokaryotes. And remember, that's actually a really good point. Operons, this doesn't happen on our cells. This is for prokaryotes, which is something that you might think is confusing because some people are like lactose intolerant. So you might think that the operon is something that works in us, is something we have. But that's completely different. The way you can kind of think of it is that ours is more complicated. And so we need like transcription factors, and we need a lot of different other stuff like enhancers that's more specific. And so, yeah, just keep that in mind. But basically, the other thing that's going on is that CAMP, which you might have remembered from cell signaling, serves as a kind of molecule that can bind to this protein called CAP. And once it's bound together, the complex sort of acts like a transcription factor, but not really because prokaryotes don't have transcription factors. But this complex will bind to the promoter, will be on the promoter, and it'll increase the transcription of these genes. So basically, you can kind of think of it as it going to the promoter and then signaling to the RNA polymerase, hey, like we need to create these genes. So come down and help us create these genes, which increases the chance that these genes are going to be transcribed. So yeah, and that's in the presence of only lactose. And now when we go on to the presence of glucose, then you'll see how this process is different, especially the CAMP and CAP. Yeah. So I'll give you a minute if you want to ask any questions, and then we'll move on to glucose and lactose, or glucose presence. Yeah.
The chat is
We got a question. How does glucose stop the process of making CAMP? Which is a really good question. Now that we're moving on to the presence of glucose, we can talk about that. Just give me 1 second to erase the board. Okay. This.
Erase your enzyme names.
My
Yeah,
enzyme names?
Yeah. You don't need those.
Okay.
I think.
Okay. So, specifically, when glucose is present, it acts as. I'll draw the picture. That's it. It's like a little lima bean. Right. So if you remember from cell signaling, oh, my bad. If you remember from cell signaling, we have this enzyme that is called the adenyl cyclase. And what that does, based on the name, adenyl cyclase, the ACE, obviously enzyme, cyc, that means that it's going to make something cyclic, right? Adenyl is just the nitrogenous base that it's working with. And so what it does is it takes ATP, cuts off two of the phosphates that ATP has, and then bends that molecule to make a cycle. And so that's how you form cyclic AMP, right? Cyclic adenosine monophosphate. That's what CAMP stands for, just in case that wasn't clear. So it's a cyclic molecule of AMP which is just adenosine monophosphate. That means it's a nitrogenous base with one phosphate that's bent into kind of like a circle kind of shape. So the reason why I brought this up is because when glucose is present, it acts as a sort of inhibitor to this kind of reaction. And so when we have glucose, yeah, when we have glucose present in the cell, it kind of shows up and it's like, hey, party's over, don't no more. And so this reaction, so this conversion isn't done anymore. And so therefore your ATP isn't being converted to CAMP, right? And without the CAMP, then this cap, since for this to act as a transcription factor, it needs to have the CAMP and the cap protein bind together. But in this case, when there's glucose present, there's no CAMP being created, and therefore you can't form the complex, and therefore it's not going to bind to the promoter and act as a transcription factor. In addition to that, we can talk about the rest of this, how the glucose affects the rest of the operon. But let me know in the comments if that answers your question. Let me write down some stuff first. Okay. What else was I gonna say? Oh,
Can you jump to like the whole point is to save energy?
yeah. I was going to talk about that
Okay.
at the end.
Yeah, you said you got it.
Cool. So in this case, we're talking about in the case of glucose presence and no lactose present. And so when we have glucose, right, you got to remember what the purpose of this operon is. Like I said in the beginning, the purpose of it is to take this molecule of lactose, break it, and get the glucose that we want, right? Because that's really what we want from lactose. We don't really care about galactose or anything like that. We want glucose. And so if the cell has glucose, right, then what's the point? Even if in this case, we're not talking about with lactose, but if there was lactose, there's no point in dealing with that lactose because we already have what we want, which is glucose. And so in this case, when there's no lactose, that means there's no allolactose that's going to bind to here. And therefore, this repressor is going to be active, and that way it binds to the operator, and that way this whole operon is off, right? So the way you can kind of think of it is that, actually, yeah, we'll take a break there. I'll give you a minute to kind of process it, and let me know if you have any questions. You can put your question in the comments.
All right, I love all of you, but I'm not staying
Oh.
now.
Thank you, Gary.
All right, so we're back.
Yeah.
So now we're gonna go through the process when there's both glucose, that one, and lactose. So let me erase this. I'm gonna erase this. I think that's yeah. All right. So so we have glucose and lactose present, right? So remember, the purpose of this operon is to break down lactose, right? So, but if there's glucose present, then you don't really want to deal with lactose. But so what happens in this is kind of a little bit more of an interesting case because you have them competing against each other. So what happens is when glucose is present, you want that, right? You want that as your energy source. So what that's going to affect is this right here, the process of that adenylyl cyclase does by converting ATP to cyclic AMP. That's what glucose, the presence of glucose is going to affect. So basically, because glucose acts as an inhibitor to this enzyme, you're not going to see ATP being converted to CAMP, right? And so because there's no CAMP being converted or CAMP being created, you don't have this transcription factor that's made out of the CAMP and the CAP binding together to become an active transcription factor. You don't see that, and so you won't see an increase in transcription for this operon. And then on the other hand, though, when you have lactose present, like when we talked about in the case that we first talked about. In the first case that we talked about, allolactose, right, serves as a sort of molecule that will bind to the repressor of this operon, and it will make it inactive. And so this repressor won't be able to bind as nicely to the operator, right? Because you have this puzzle piece that's like this, and now you used to have a repressor that was like this, and it could fit perfectly, right? But now you have it like that, because now it's bent, and so now it can't fit perfectly into this operator. And so, in a way, you increase the operon being transcribed because you don't have a repressor anymore. But at the same time, you don't have CAMP binding to CAP and creating that kind of transcription factor that would increase transcription. And so, even though there's like a sort of, it seems almost balanced, we're going to call this operon off. The reason being is even though it seems kind of balanced, like, oh, the repressor being inactive, well, that would increase the transcription, but the CAP and CAMP is not being created, or the CAMP is not being created, you don't have an activator. And so it seems like almost it's balanced, But if you remember back to what the purpose of this operon is, you want to use glucose, right? You want to break down lactose, or the purpose of it is to break down lactose and use the glucose that the galactose has. And so because you don't have, or because you already have glucose, you have to think of it in terms of energy, right? If you have, let's say, no, that doesn't stick. Okay. If you have glucose and then you have lactose, if you have glucose, you can automatically use it. You can just automatically use it, take it, right? You can, and then use it as an energy source. But if you only have lactose, well, if that's your only energy source, then you need to break it down, you have to break that bond and get that glucose that you want. But when they're both present, well, why would you go through the hard work of breaking this apart, getting rid of like the lactose, and finally getting this glucose if you already have glucose in the cell? So when you have glucose and lactose, you're going to favor glucose. And that's why this operon is off, because if it was on, then you would be favoring lactose and you'd be wasting energy. And so you don't want to waste energy. Now, the way you can kind of summarize this whole thing is, actually, I'll give you a minute to kind of digest this. Ask any questions in the chat. And then we'll move on to a kind of, what's it called?
Graph.
A graph. Yeah, to kind of see how the different levels of on or offness. Yeah, we'll start.
Can we just use those?
So that's a really good question for what happens to galactose, because we kind of ignore it when we're going through it because all we want is glucose. But if you remember, galactose is a sort of isomer of glucose. And so if you have galactose, if you have the molecule of lactose, on my nifty demonstration, if you have glucose and galactose, this is lactose, right? If you have glucose and galactose, you want this glucose. And so you'll be able to use that directly and use it as an energy source. But you're left with this galactose. Well, instead of just kind of just throwing it away, you can convert this to glucose. And now you have two glucose molecules. But then again, that conversion, right? You have to first break it. Now you have glucose, but now you have to change this one to glucose as well. And so based on all that, you can start seeing why it's a very energy dependent process. It needs a lot of energy to break it, and then you need to convert this one. And so that's what happens. For the concept of this, in the context of this class, you don't need to know what happens to glucose. That's more like a biochemistry thing, which you'll learn about in upper division. But yeah, that's what happens to galactose. So we can move on. Let me know if that answered your question. Let's move on to talking about basal level expression versus elevated or high levels of expression, and because most students kind of get troubled by those terms. I can erase it,
Animation.
Hmm?
Alright, you're back on.
All right. So, okay, so the graph is
You said thanks.
like level of lactose. No lactose. Maybe I should start with that.
You should, yeah.
No lactose. Lactose. Lactose and glucose.
Yeah. And then also, do you need just glucose?
So, and then this will be what we consider basal. So before we go into all these types of scenarios, we need to talk about what this means, basal level, right? So basically the word basal just refers to the fact that when you don't have a lot of lactose present, then you still might have some level of expression, which seems kind of weird because we just went over how, when you don't have lactose, why would you want the operon on? But the way you can kind of remember, or the way you can kind of think of it, is that that repressor. I should draw it again. Remember, this repressor doesn't have anything to do with this operon, right? It's not considered a part of the operon, but this repressor is always going to be made, always, 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 always. And the thing is, when there's no lactose, like we said before, this repressor is going to go and bind to the operator. It's going to bind to the operator, right? And it's going to say, hey, RNA polymerase, no transcription, please. And so the thing is, though, in biology, there's not really definites. And so even though this repressor is bound to the operator, it's not going to stay on forever. It's not like a very strong grip where it's going to stay there forever. And so sometimes, once in a while, it'll get tired and it'll break off. So, there's nothing wrong with it. It's still active. It's just that, it gets tired. It gets tired of just hanging out with the operator the whole time. And it, takes a small jog or whatever. So in that time, the RNA polymerase is like, oh, it's my time to shine. And so it's like, okay, the repressor isn't there anymore. I'm going to go sneak in really quickly and transcribe these just because there's no repressor, right? But as soon as a repressor goes back on, transcription is stopped. No more transcription, right? And the RNA polymerase can't just sneak in anymore and do transcription anymore. And so that's going to happen very frequently. And so that's what we call basal level expression, which is this right here, where it's not really no transcription, right? So this would be this right here, the x-axis, that would be zero transcription, right? That never happens.
It's like, yeah, it's like absolute zero.
Yeah, absolutely zero, no transcription, which isn't what happens. Does that make sense? Yeah, English. So what happens is what we talked about, where this repressor will get tired, it'll latch off, and you'll have a little bit of transcription here and there when the repressor kind of gets tired and kind of goes away from the operator. And so that's what we call basal level, okay? And even though that's like not zero transcription, we consider this off. Yeah. So, and then this is high. Can you see the arrow on the side? Okay, cool. So we have this basal level of expression, right? That's when we have no lactose present. So there's no reason for this operon to be on. So it's off. And also this repressor is going to get bored. It's going to get tired. It's going to latch off, right? And so we can have some transcription. And so when we have no lactose, we're going to have basal level transcription, which all this something I forgot to note is that basically we have this high bar that we have to cross in order to call the operon on. So if there's very, very high levels of transcription or expression, then we consider that on. Anything below that is off. So even if there's elevated transcription, like a medium level, that would be considered off. And if there's basal, then that would be considered off. So all this right here is considered off. Anything above high levels of expression, that's on. So now in the case of lactose, we do have in the case of lactose, we'll have that we'll have the repressor inactive. Inactive. And then we'll have CAMP, CAMP complex. In the case of lactose being present, we'll have the CAP and the CAMP complex put together, and so that's going to act as a transcription factor, and that's going to elevate the transcription, as well as in allolactose turning off the repressor, right? And so that allows for the level of transcription or the level of expression for lactose, when there's only lactose present, to be high enough that we consider the operon on. Now, when we have the next case, when we have lactose and glucose, that's when we go into the case where we don't have a CAP. We don't have that CAP CAMP complex, right? And the reason why is because we have adenocyclase being inhibited by glucose being there. And so you don't create CAMP, and therefore you don't create that complex of CAMP and CAP. And so it's not completely basal. So that's what we would consider medium, right? Well, because we have the repressor off, or we have the repressor inactive, there is a bit of elevation because we don't have this thing telling RNA polymerase not to bind there. But on the other hand, we don't have that CAP, CAMP complex telling the RNA polymerase, hey, come here to transcribe this. So it's not completely on, it's not completely off, and that's why we consider it medium. But because like we said before, it's not passing that high bar, right? It's not high levels of expression, then we don't consider it on. So, like I said, so this would be off, this would be on, this would be off, and basal. Yeah. So just to review, no lactose, it's off and it's basal. Lactose would be on because there's a high enough level of expression that's going on. And lactose and glucose would be off, but it wouldn't be at basal level because we have the repressor inactive, but we don't have the active CAMP complex. Now, for the last case, glucose. So when we have glucose present, that would mean that we have this is the case when there's only glucose, no lactose, right? So that means that the repressor would be okay. So when there's only glucose, there's no allolactose to turn off this or to inactivate the repressor. And so the repressor is going to be able to bind to the operator, right? and there'll be an inhibition of adenyl cyclase, and so the CAMP won't be converted, or the CAMP won't be created, and therefore you don't have that transcription factor that will elevate it. And so in this case, it's kind of very similar to the no lactose case, because you have the repressor off, or the repressor on, and you don't have the complex. Actually, no, just kidding. You have the repressor on or active, and you don't have the CAMP complex. And so this would be, basal, similar to no lactose. But like we said, anything below this high bar, this high level of expression bar, is off. So in the case of just having glucose, it would be off. Yeah. And so those are all the cases. You can talk about, and this one it's no glucose. No glucose. Yeah. So these are all the 4 cases that you'll learn about in this class, and then you'll expand on them later on in biochemistry, particularly in the case where, let's say, you run out of glucose and you have lactose left. And then let's say, like, oh, what if there's high levels of glucose and low levels of lactose, or there's high lactose and low glucose. And so you'll get into more details when in upper divisions. But this is basically the summary of the lac operon. So yeah, I'll give you a minute to kind of digest it, and then post a question in the comments if you have any questions at all. If not, we'll move on to trip operons. Yeah. By the way, if you haven't signed in yet, if you haven't signed in yet, the link to sign in is in the comments. Just scroll up, make sure to sign in. But before we move on, Derek brought up the good point that even though this is a very useful thing to kind of distinguish the cases, in this class, really, if you talk to Dr. Latham, there's no such thing as medium. It's just a thing that we kind of talk about to kind of visualize it a little bit more. So it's not like a technical term. It's only really basal or high that we really use. And in addition, even though I drew this medium to be kind of in the middle, I'm going to draw a different graph that's going to show you the bigger difference between them.
Let's see. Wait, just see level of expression.
Level of it. Yeah.
Yeah, look at my next.
Oh, gotcha. Okay. So, basal.
Yeah.
Medium. So, when we talk about basal, right, we said that there's very little transcription that's going on, right? Because we have the repressor bind to the operator, but then it's like takes a break, gives it a second, and then it binds back again. And in that time, the RNA polymerase is going to bind and do some transcription, right? So it's a little bit of transcription. It's not zero, right? Basal is not zero. It's very low. Now, when we talk about medium, that is an elevation from basal, right? But it's really good. When we talk about medium, it's a little bit greater than basal, right? It's a little bit higher than basal, but at the same time, as you guys can see, it's not that much of a difference. But when we talk about high, then it starts looking like there's a big difference, right? So high would be a very high level of expression because, like we said, yeah. So as you guys can see, even though we have medium, it's not really that much compared to basal, and compared to high, it's basically almost basal. And so that's why, really, in the terms of this class, and usually in terms of any upper division, we really just talk about basal and high because medium isn't that much higher than basal. But it's a specific case where you have glucose and lactose and they're competing against each other. That's when we use medium, but then again, it's not that much higher than basal. So yeah, we can move on to trip hop run. There. So what I drew is the tryptophan operon. So basically, tryptophan, TRRP. Oh, I don't know if you can see because of the glare, but it's supposed to say the TRP stands for. Oh no, now you can't see it. You can see it better. I don't know. TRP. What it says up here is TRP, tryptophan operon. So like the lac operon, it's a collection of genes, right? A, B, C, D, E, that are useful in the creation of, or they're all controlled by by a common promoter and operator. And so the difference in this one is that for tripoperon, the tripoperon, instead of breaking down tryptophan, like how lac operon breaks down lactose, tryptophan operon actually creates tryptophan. And so when it comes to the tryptophan operon, for prokaryotes, it's basically the purpose of the tryptophan operon is to create tryptophan because it's a amino acid that most organisms kind of lack, and so they need to create that amino acid. And so if you think about it, this operon does that. And so basically, we're going to look at the case where there's no tryptophan. No tryptophan. And so what happens is, similar to the lac operon, it's going to have a repressor that's not a part of the operon that's always going to be made. But in this case, in contrast to the lac operon, the repressor is going to be inactive when it's created. And so the reason why that's important is because when there's no tryptophan, right, when there's low levels of tryptophan, well, that means that you want to create tryptophan. Because your body needs tryptophan, and if you don't have any, then you want this operon to be on because you want to create tryptophan. So when there's no tryptophan, the operon is on, and that is why this repressor is going to be inactive, and therefore it's unable to bind to the operator, and therefore the RNA polymerase is able to bind to the promoter and then transcribe all these genes. Another thing is that these genes, A, B, C, D, E, they're not really important in the terms of like you need to know what they code for. They're just genes that will eventually create tryptophan. And so, yeah, so because the repressor is inactive and unable to bind to the operator, transcription is high, and the operon therefore is on. So now let's talk about, let's say this keeps on going, right? Let's just say the operon keeps on going, it's on, it's creating all this tryptophan, right? Eventually you'll have enough tryptophan in your body to do whatever you needed to do with the tryptophan. And so in the case similar to lactose, the thing that turns on this repressor concentration. When there's high levels of concentration or high levels of tryptophan present, then it's going to act as it's going to bind to the repressor. The tryptophan is going to bind to the repressor and it's going to make it active. And when it's active, then it's able to, because it's a more nicer puzzle piece, it's able to bind to the operator. And when it's bound to the operator, that doesn't allow RNA polymerase to do transcription of this. which makes sense because, like we said, the purpose of this operon is to create tryptophan. And if you have high levels of tryptophan, then you don't want this operon on anymore because you're just going to waste energy because you already have enough tryptophan made. What else? Yeah. Oh, and then so in the concentration, in the case where tryptophan is high, a high concentration of tryptophan, operon is off. Yeah. Much more simple than lac operon. Yeah. There's not really anything. There's not really anything else. It doesn't go into as much detail as the lac operon is, with the whole cap and the glucose being present and competing. In this one, you just need to know the two cases where there's a lot of tryptophan and there's a low amount of tryptophan. And if you just remember the purpose of the operon, you're able to tell. When it's supposed to be on and when it's supposed to be off, because you know that when there's high levels of tryptophan, you don't need to make some more, and the purpose of this is to make more. And so if you already have high levels, then you don't need to make more, so the operon is off. If you have low levels and you need to make tryptophan, then this has to be on so you can meet those levels of tryptophan that you need for your body in forming proteins, because like we said before, tryptophan, TRP, is an amino acid, and that's what makes, or it's once you put amino acids together, you make a protein. So yeah, I'll give you a minute to digest that and see if you guys have any questions about this. And don't forget to sign in if you're just joining us. So yeah, after this, we might go into mitosis or meiosis. Or if you guys want to talk about lac operon again or trip operon, just if you want to go through it again, just let me know. But put like a little comment in the chat, and I'll give you a minute. So yeah, if you guys want to ask, if you guys want to go over anything, post the questions because we'll be ending the stream pretty shortly. But before we do that, I don't know if there's any new viewers or anything like that. We can just go over the operons again, or have like this little summary chart so you guys know when it's on or off. I don't want to start meiosis or mitosis because we only really have 30 minutes left. And you guys barely started covering it yesterday on Wednesday, and you're going to cover it in more depth on Friday. So we'll leave that for next week. And if you have questions, post them in the comments. But right now, I'm just going to make a quick chart to explain or to summarize this whole CSS. Yeah. Oh, and while I do that, post any and all questions in the comments. Okay, so this is kind of like a summary chart of what we went over today. So we'll start with the lac operon, and as I go through it, quietly, or if you want to post in the comments what your answers might, or what you think, if the operon is going to be on or off, and we'll go through it, or you can shout it out in the comments. So for like we said, for glucose and lactose being present, remember that's like the medium, right? But remember, because medium is not that much elevated compared to basal, we're gonna consider that off. I don't know why I waited for somebody to say something. All right, so we're gonna consider that off. When there is no glucose, that's what the minus sign here means, but there is lactose present, that's when you have the repressor being inactive, and therefore, and CAMP and CAMP and CAP complex being on or activated, and therefore you have the operon being on. Now, when we don't have lactose, but we do have glucose, that's when you have the repressor being bound to the operator, therefore no transcription, and you also have the inhibition of adenyl cyclase, and so you don't have the CAP and the CAMP, and so that would be off. Oh, should I write that in there? I should write that in there. Let me make these a little bit bigger.
Which one you
What's up?
got? What do you got?
I was gonna write what's gonna be.
Like level?
No, not level, but rather what's on and off. So this one was off. Inactive
Oh, repressor.
inactive
Cap.
Oh, I should remember it better. Inactive cap. This was on, and this was inactive. Active. Yeah, and then now when we're talking about the case where there is no lactose present and there is glucose present, that's when we don't have. That's when we have the active repressor and we have the inactive cap. And so you have the repressor telling the operon to don't do transcription, and we have the inactive cap telling not the inactive cap not promoting transcription. And so that would be off. Now for this one, when you don't have either lactose nor glucose, that's the case where you have the repressor is active. Just gonna abbreviate it as rep active. And you have the cap.
It's irrelevant.
Okay. All right. Yeah, because I don't think we ever talked about that. The important part is that when you don't have glucose nor lactose, is that the repressor's on, or sorry, the repressor's active. Try to avoid saying that the repressor's on or off, because that might confuse you when you're thinking about, oh, is the operon on or off? So when you're talking about, when you're telling, or when you're trying to determine whether the operon is on or off, look at whether the repressor is inactive or active, and if the cap complex is inactive or active. You don't want to say on or off because it gets confusing. So this one would be off, because you have the repressor active, and so it's binding to the operator saying no transcription. And now we can go through the trip.
Well, for the no lactose, no glucose, there's no point to make cap because, well, cap is to breaks down stuff for energy. You have no energy source triggered.
Yeah.
So
should I put inactive?
I would just put lines with it. It's like, yeah.
Okay.
For the sake of continuity.
Not applicable. Yeah. All right. So now when we move on to the trip operon, we have the case where there's, I should do, yeah. So when I say plus TRP, that means there's high levels of tryptophan. And so remember, the trip operon, the purpose of that is to create more tryptophan. And so when there is tryptophan, when there's high levels of tryptophan, then you don't want the operon on, because you're going to waste energy creating something that you already have. And so that's the case where you have. That's the case where you don't want the operon on, you want it off, and that's the case where your repressor is going to be bound to the repressor is going to be bound to the. And so when we're talking about the case where there's low tryptophan concentration, that's the case where you want the operon to be on, because hey Emmanuel, you want the operon on, because when you have a low level of tryptophan, you want to use that operon that you have to create more tryptophan, because you might need tryptophan to create proteins and all that other stuff. And so in the case where there's low tryptophan, you're going to have the repressor being inactive, because there's no tryptophan to bind to the repressor, and that makes it active. And so that's when you have the case where it's on. And so yeah, that's basically a summary of the lock operon and the trip operon. We can run through. How much time do you have left? Okay. Post any questions in the comments. We're gonna probably end around a little bit before 8. Just for right now, I can just quickly, I guess, go through some differences between them. Yeah. Yeah. So while I do that, post comments about any questions, whether it's about this or anything else in the course. Is this still on the screen? Needs to see that?
Yes.
Yeah, okay.
Oh,
Yeah.
constituted and inducible.
Inducible?
Well, no, they're both constituted. Their structural genes are inducible. Yeah, don't worry about it.
Okay.
It's just vocabulary later.
Yeah.
They're not.
What else was I going to say? Purpose to make. TPR. And then this one is purpose to break. All right. So, also, what they have in common. Oh shoot, where should I write that? Where's space? Oh, okay, there.
Right. Okay.
Am I still on screen? Yeah, a little bit. Oh, there we go. All right. So before we end the live stream, the last thing we will say, or the last thing I'll say, is that for the LAC operon, the difference between the LAC operon and the TRIP operon is that the LAC operon, the repressor is actively made, and so when it's made, it's active, and compared to the TRIP operon, when the repressor is made, it's made inactive, and so it needs to be turned on, or it has to become activated. And then the other thing is that the purpose of the LAC operon is to break down lactose, while the purpose of the TRIP operon is to make tryptophan. And so one is to break, one is to make. So that can help you remember when it's going to be on or off. And the last thing is that both these operons in general, those are going to be found in prokaryotes. We don't have that, even though you might think that lactose, some people are lactose intolerant. You might think that, LAC operons are in us as well, but it's not. It's a completely different process. But yeah, that's about it. Glad you stayed tuned. Make sure to sign in if you haven't already. One sec.
Inducible versus always on. You can turn on.
You're right. Yeah, so really quickly, before we end, we can go over the last bit of terminology.
You just say it.
Okay. Can I see the words again? Yeah.
Inducible or
All
constituted.
right. So, where is it? Right here. So if you look right here, inducible versus constitutive. So inducible, that means that you can induce something to be on or off, while constitutive means it's always being it's always being done. So in the example for constitutive in this in the context of operons is a repressor, because the repressor is being made all the time. Now, is the repressor active or inactive? Well, that depends on the scenarios that we have, right? Lactose present, glucose present, stuff like that. And inducible is things that you have to turn on, or you have to make active, or you have to do something to it for it to be expressed. And in that case, you can talk about the operons in general, right? because these operons aren't just going to be on regardless. They're not going to always be on. That would be constitutive. The repressor is always going to be made, but the lac operon and the trip operon aren't always going to be on, and so they have to be induced or forced to be turned on. And so yeah, just a couple of terminology that you might hear when we talk about it a little bit more, and also in upper division biology. So it seems about time. If you guys have any questions, please post them in the comments. Make sure to like and subscribe. We're gonna try doing these for the rest of the semester, hopefully every Thursday. And if, like as always, they're gonna be posted right afterwards. So if you missed anything, or if you have friends that are like, "Oh no, I totally forgot to sign on to CSS on Thursday," tell them that it's gonna be on this YouTube page, and yeah, I'll wait for a minute or two before we end, just in case
Oh, we
there's
got
any
something.
last thing.
Both
Oh,
operons
just-
are positive and negative regulation.
That is
Positive regulation is just an activator. So the lac operon is both. Trip operon is just negative. Because negative is just a repressor.
yeah, and because the lac operon has the
It's cap.
cap and the repressor. Yeah. So yeah, like Derek pointed out, that was a good question because we didn't really go into positive or negative regulation. But for the lac operon, as you can see, there's we're talking about cap and the repressor. And so the repressor, that's negative control. And so the way you can kind of remember it is that negative.
It's like a brake, gas pedal.
Yeah, gas pedal and brake. So the repressor, that's the brake, right? So it forces the thing to stop, while the positive regulation is something that forces go or to be
Increased, right?
to be increased. And so you can think of that as the gas pedal. I was going to say throttle for some reason. So the positive control would be the gas pedal, and then the lac operon's case, that would be the cap when it's on, and then the negative regulation would be repressor, whether it's on or off, and how that affects it. However, in the case of the trip operon, that would only be negative regulation, because you're only really looking at the repressor. There's not like a cap or a CAMP cap complex that acts as a transcription factor like you see in the lac operon. Yeah, that was a good question. I think, any other questions? I'll give it a minute or two. If not, then we'll end the live stream. But yeah. So I think Yesenia, I think you're the only one on the on the live stream. So if you have any questions, maybe post them on the comments. I can stay for another 5 minutes. If not, you can just tell us that you're good, you understood everything, and then we can just end the live stream. Or you can tell your friends to hop on in the last 5 minutes if they have any questions. Yeah, but we'll be here for a bit more.
She says she's good. Thank you.
Oh, okay, cool. Yeah, so I guess that means that's it.
Yeah.
Yeah, so like I said before, like, subscribe. We'll be here every Thursday. Tell your friends, Yesenia. Tell your friends. And yeah, let me know if you really enjoyed this by giving me a like or telling me in class. And yeah, all right, we're good.
Signing off.
Signing off. Okay, end stream. Am I sure? Yes.